Yeah. So this information cannot be just stored for very short time because you need to pass it down. So until you reach maturity, okay, this information can be passed down and it's need years and years, all right, and able to replicate accurately. Just imagine these informations were not accurately or inaccurately passed down to the next generation. What will happen? Yeah, what will happen if your DNA was inaccurately passed down to the next generation? Okay, so there's a huge, very serious complications will happen. All right, and also it's capable of change to allow evolution. Yeah, all these informations, they allow evolution. So in the early of 1990s, the chromosome was shown to be the carriers of hereditary informations. So in eukaryotes, they are composed of both DNA and proteins, and most scientists initially believe that protein must be the genetic material. Okay, because they only because they and that moment they still don't know what's gene. Actually, genes actually encode for the protein, but at some point they only believe that that's only the protein. So in search for the genetic material, many early experiments were conducted. Okay, such as the Griffith transformation experiments. Every transformation experiment, they use the bacteria and also the phage, you know, and also Hershey's bacteriophage experiment. Okay, these are the three very well-known experiments to a very early era of searching for genetics material. So these are three very important experiments which I need you to do. Okay, so this is come to the second assignment that you discuss in detail the principle and findings of these three early experiments in genetics. So I posted this in Ellipse and together with the instructions, so you can do it. Okay, this is for your second assignment. All right. So there are three, which is the Griffith transformation experiment, the average transformation experiment, and also the Hershey-Chase bacteriophage experiments. So, clear with this?
Yes, doctor, clear.
Okay, good. So, I have just decided we probably will have 6 assignments. Okay, 6 assignments, and because it's very important, you are not coming to university, so we need to give you more assignment. Okay, so that you can understand more. When you read more, when you do the assignment, you actually will understand more. All right, because you don't come to, because we cannot see each other physically. So this is the only way that we can help you, okay, and force you to read more. All right. So now, what is the compositions and structure of DNA and RNA? I believe you know what is DNA and RNA, right? So basically, DNA and RNA are polymers that compose of monomers, what we call the nucleotides. Okay, and then each nucleotide has 3 parts. The first one is a pentose 5-carbon sugar. Pentose means pen. Pens mean 5. So there are 5 carbon, okay, sugar, okay, as anchor, and they also have the nitrogenous bases, which is the ATCG or AUCG, and a phosphate group. Okay, that you can see this one. Can you see this?
Yes,
okay,
doctor.
that's the nitrogenous bases. Okay, and you have the sugar, the pentose sugar, 1, 2, 3, 4, 5 pentose. This is the 5 carbons here, and also one of the phosphate group. Can you see my laser pointer?
Yes.
Yes.
Okay.
Yes,
Good.
doctor.
Yes,
All
doctor.
right. So the pentose sugars in RNA is a ribose. It's different because in DNA it's an deoxy, but in RNA it's a ribose, and it is in DNA is what you call deoxyribose. So you can see the difference. The only differences, okay, the only difference is the second position here. The second position you can see, yeah, position 2, carbon 2 here, where the RNA has a hydroxyl group, okay, which is OH group, while DNA has only a nitrogen. You see the difference?
Yes, doctor.
Yeah, the carbon 2 here. So that is why RNA is what we call ribonucleic acid, whereby DNA we call it deoxy, whereby one oxygen is gone at the carbon 2 here, which is replaced by hydrogen, whereby in the RNA they attach with the hydroxyl group. Okay, that is the major difference between RNA and DNA. Okay, so far okay?
Okay, Tutor.
Yes.
Yes.
Okay, so
Yes,
what is the three main components of RNA and DNA? Huh? Okay.
Nitrogenous base.
Base.
Okay, nitrogenous base.
Phosphate group.
Phosphate group. All right. So it's very important for you to know this. And the only difference between RNA and DNA is at the carbon 2, whereby RNA has hydroxyl group and DNA has hydrogen. Okay. Is that clear?
Yes.
Okay.
Yes,
Yes.
doctor.
So there are two classes of hydrogenous bases. One is what you call purine. One is called pyrimidine. Purine has double ring, okay, whereby the structure have 9 members, include adenine and guanine, okay, because they have this one, okay, two rings, right? 2 rings, okay. This is purine. This is the parent component. They have 9 members, whereby 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are 9 members. So if you look at the adenine here, the carbon 6 is replaced by amide and NH2, whereby in guanine it is replaced by oxygen. Okay, and in guanines the H2N is attached to the carbon 2. Right, so the major difference between purine and pyrimidine is that purine have double ring, whereby pyrimidine has only 1 ring. Purine has 9 members, whereby pyrimidine has 6 members. And these pyrimidines includes cytosine, thymine, and also uracil in RNA. Yeah. Can you see that? Okay, pyrimidine, they only have 6 members. Okay. All right.
Okay,
So you
doctor.
can see cytosine. The differences between here is at the carbon 4. By thymine, they have the CH3, which is the methyl group, at carbon 5, whereby uracil they don't. They only have H. Okay, H. So you can see also the differences between RNA and DNA is that in RNA, they are called AUCG, whereby in DNA, they call ATCG. So thymine is replaced by uracil in RNA. That's the main difference also between RNA and DNA. Okay?
Clear,
Clear?
doctor.
Okay.
Yeah.
Right. So the structure of the nucleotide has these features. First, the basis. Okay, it's always attached by the covalent bond between the first carbon. Okay, first carbon of the pentose sugar and a nitrogen in the base. Okay, so specifically the 9 nitrogens in the purines and 1 nitrogens in the pyrimidines, as you can see here. All right, this one, this is the carbon 1, right? You see, can you see this? So
Yes.
it's attached to the bases. Right? Boleh ya?
Yes, doctor.
So you see, nucleotide is the sugar plus bases plus phosphate group. All right. So deoxy adenosine, they only have the monophosphate. So the sugar-base combination is the nucleoside. The sugar and base, which is the pentose, and base, which is the ATCG or AUCG, they call nucleoside. Okay, when a phosphate is added, okay, always to the 5' carbon of the pentosugar, sugar, then it's become the nucleoside phosphate or simply nucleotide. So you can see now the differences between nucleoside and also nucleotide. Okay. Clear?
Yes.
Okay. So
Yes,
nucleoside
doctor.
is without
Without phosphate.
yes, without the phosphate group, whereby the nucleoside is the combinations of sugar, pentose, and also the bases. Okay. Now, so the polynucleotides of both DNA and RNA are formed by a stable covalent bonds, which is a phosphodiester linkage, okay, between the phosphate group on the 5' carbons of one nucleoside or nucleotide and the 3' hydroxyl group on another nucleotide. So these create the backbones of the nucleic acid molecule. As you can see, okay, this is the phosphodiester bond. Okay, and this is the hydrogen bonds. You see that? Okay, so the
Yes, doctor.
asymmetry of the phosphodiester bond create the 3' to 5' polarity within the nucleic acid change. So you can see the nucleic acid is double helix, isn't it? Yes.
Yes.
It's
Yes,
a double
doctor.
helix structure, and one's from 5' to 3', another one is 3' to 5', right?
Yes.
So when they are coiled together, that you can see they are very stable. Yeah, because the one from 5 to 3, the other one is 3 to 5. And they form a very strong phosphodiester linkage or phosphodiester bond. Okay. So DNA must be very, very stable. If they are very fragile, then it's very dangerous. Yeah, because they cannot be, they cannot produce a protein which is needed by our body, or the necessary enzymes or hormone that are needed for the physiological processes. Okay. So you need to see. Okay, this is the phosphodiester bond where they create, okay, where they link it together, and also the hydrogen bonds. Okay, boleh ya?
Boleh, doktor.
Yes.
Okay, this one
Boleh,
also
doctor.
hydrogen bond. Okay, this one is the, how about this one? This one is,
Phosphodiester.
yes, phosphodiester bond. Okay, very good. Sangat bijak you all. So when Watson and Crick, okay, they begin the work, has known that the DNA is composed of nucleotide, but how the nucleotides are assembled into nucleic acid was unknown. So there are two additional sources of data assisted Watson and Crick with their models. For the first one is the Irwin-Chargaff's ratio, where they obtain the DNA derived from a variety of sources, show that the amount of purine always equal to the amount of pyrimidine. And further, that's the amount of G equal to C and the amount of A equal to T. Yeah, you know, this is why A is the one that attached to T, and the G always linked with C. Yes?
Yes.
So, so when you want to find out the amount of C, you just need to look at the amount of G. Correct?
Yes.
Yes.
If you want to look for amount of A, you always look for amount of C, correct?
T.
Yes.
Oh,
T.
Good. At least you understand. Huh? I'll try to give you the wrong fact, and I want to see whether you really understand or not, because some people may have fall asleep already. Huh? So A always equal to T and G always equal to
C.
C. Okay, so the amount must be very similar all the time. Right. So this is the first clue that Erwin Chagall provide the proof to Watson and Crick for the double helix model. Another one, very important one, is the Rosalind Franklin's X-ray diffraction. Images of the DNA show the helical structure with regularities of 0.34 nanometers and 3.4 nanometers along the axis of the molecule. So many people say, actually, Rosalind is the one that found out the double helix. But too bad, because she died, right? Because she died earlier. That's why the credit was taken by Watson and Crick, and some people, claim that Watson and Crick actually steal the information from Rosalind Franklin for him to, came up with the double helix model. I don't know whether you know this story or not. Have you ever heard of this?
No.
No, doctor.
Yeah, because Rosalind died earlier because she's doing X-ray. Yeah, and last time the safety features are not so good, so she exposed a lot of X-ray and she got radiations. Okay, very badly affected her health, and she died at a quite early age. And her contribution was taken credit by Watson and Crick to come up with the double helix models. And at the end, Watson and Crick won the Nobel Prize. Okay, but we still acknowledge Erwin Chagall's and Rosalind Franklin's research, because without these important points, Watson and Crick will never be able to come up with the double helix models. Okay.
Okay, doctor.
Okay. Now the DNA double helix. We all know that our DNA is a double helix. So what is double helix? There are three main features. Okay. So it is two polynucleotide chains wound around each other in a right-handed helix. So you can try to draw that. Take two pencil, okay, and tap together, and then you can draw something like this. Okay. All right. You can try to draw. You can get a red pen or a blue pen, and then you tie it together, and then you try to draw the double helix. So one is running from 5' to 3', another one is running from 3' to 5'. And you can see that here. So in this double helix, the base is actually right inside here. Okay, and there are, okay, the bases here you can see, okay, the base pair. Okay, and this is the phosphodiester, this is the bond, the phosphate group, and this is the minor groove. Okay, minor groove. And this is the major group. Okay. So if you look at that, so every 10 bases, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So they make 3.4 nanometers. So that means the differences between each bases is 0.34 nanometers. Okay. So this information was provided by who?
James Watt.
Erwin.
Huh?
Erwin Chagall.
Franklin.
By Rosalind Franklin. Okay.
Oh, yes, yes, yeah.
Yeah. Can see this?
Yeah.
Yeah. Right. So for every 10, if you calculate the 10 bases, is about 3.4 nanometers. And this is very important. You can see the turn is a complete turn, right? From here, it's 10.
Yes.
So this is a complete turn. So the distance is 3.4 nanometers. Okay, so the two chains are anti-parallel. Anti-parallel ialah anti-selari. Satu daripada 5 prime kepada 3 prime, and another one is from 3 prime to 5 prime. Can you see that? Can you imagine that?
Yes.
Yes, doctor, can imagine.
Okay, very good. So the sugar phosphate back bonds are on the outside of the helix. Yeah, outside kan? Di luar ni.
Yes.
Okay, bases inside kan?
Yes.
Okay, and the bases are on the inside. So stack perpendicularly to the long axis like the step of the spiral staircase. Macam staircase kita lah. Ya, spiral. Pernah tengok kat staircase yang berbentuk spiral ni?
Yes, doctor.
Ya, tempat kita pijak adalah bases kan?
Yes.
Ya. Lepas tu yang tempat yang kita pegang tangan tu adalah dia punya
Sugar.
phosphate punya backbone tu. Okay? So this is what we call the DNA best pairing or Watson DNA best pairing. This one best pairing lah. A always pair with C, A always pair with T, C always pair with G. Okay?
Yes.
Boleh ya, clear?
Yes, doctor, clear.
Okay. Now, so there are 3-dimensional model has these main features, which is bases of the 2 strands are held together by the hydrogen bonds. Okay, which is the hydrogen bonds between a complementary bases, 2 for AT and 3 for GC. So if you look at C, the cytosine and guanine, they form 3 hydrogen bonds, right? Whereby A and T, they form 2 hydrogen bonds. Okay, so individual hydrogen bonds are relatively weak, so the strand can be separated by heating. For example, you can heat it up. If you want to break, if you come to 3rd year later, you have your final, I don't know, final project, or maybe you have the chance to do PCR. So you will heat up your DNA sample to very high temperature, to about 95 degrees Celsius, to break them, to break this bond. All right, to break the hydrogen bond, so that they become individual strand before you start the PCR can take place. Okay, so complementary best pairing means that the sequence of one strand detect the sequence of the other strand. So they're always complementary. Okay?
Okay.
Okay.
Boleh ya? So these best pair are 0.34 nanometer apart, and the full turns of the DNA helix take 3.4 nanometers. So there are 10 best pair in a complete turn. The diameter of the double-stranded DNA helix is 2 nanometers. So as I said just now here, dekat sini, yeah. So this is the complete turn kan? From here to here. The complete turn, you've got how many bases? Many best pair? How many best pair?
10.
10 best pair. Because the distance between one base to another is 0.34 nanometers. Okay? Clear kan?
Yes.
Yes.
So because of the way of best hydrogen bonds with each other, so the opposite sugar phosphate back bonds are not equally spaced, so resulting in a major and minor groove. So this feature of DNA structure is important for protein binding, because dia ada major group, ada minor group. Because of what? Because dia tidak equal. Okay, the backbone tidak equal, so dia menghasilkan groove. Okay, yang ada major punya dan ada yang minor pun. So this is very important for the protein binding. So supaya protein senang nak bind. Okay?
Okay.
Okay,
Kenapa
doctor.
protein kena bind? Pernah dengar tak DNA replication, DNA transcription semua tu?
Yeah.
Okay,
Yes, doctor.
untuk mulakan
Yeah.
itu kan kena banyak gunakan enzim tak?
Yes.
Ya, enzim itu adalah
Protein.
Protein.
Okay, maksudnya kalau dia ada groove, ada minor, ada major, jadi senang untuk protein ini bind lebih firm lagi kepada DNA molekul kita. Ya,
Doktor.
kalau dia jarak sama saja, maka agak susahlah dia nak bind. Terlalu licin orang kata. Sebab itulah you tengok kalau kita buat rumah, semen kita tu untuk supaya kita lekat pada dinding tu, kan tukang tu selalu ketuk-ketuk berlubang-lubang kan?
Yes.
Ya, kenapa? Supaya semen tu senang lekat kan? Betul?
Yeah.
Ya, samalah dengan major group dengan minor group ni. Supaya luas permukaan lebih besar lagi. So that the protein can bind to it easily lah. Make sense tak?
Yes, doctor.
Okay?
Yes, doctor.
Make sense kan? Okay.
Doctor, I have a question.
Yes?
How is the sugar phosphate backbones not equally spaced?
Because you see, thymine and adenine, they only have 2 hydrogen bond kan?
Yeah.
Yang ini ada 3 kan?
Yeah.
Kalau ini 2, maka hydrogen bonds tu equally spaced lah.
Okay.
Okay?
Yeah.
Tapi ini 2 dengan 3. Maka dia tidak even lah. Betul?
Yeah.
So the unevenness of this hydrogen bonds formed between the bases will result in the minor groove and major groove.
Okay, doctor, I got it.
You got it?
Yeah. Thank you.
Okay. Now, the X-ray diffraction study how the DNA can exist in different forms. So we have the A DNA, B DNA, and Z DNA. So there are different conditions. Like A DNA is the dehydrated form. Dehydrated form means air dia kurang, dia dehydrated dah. And it is so, but it is not usually found in the cell lah. So it favors the conformations at low water concentrations. And it's a right-handed DNA helix with 10.9 bases per turn, where the bases incline 30 degrees from the helix exit. And aDNA has a deep and narrow major groove and a wide and shallow minor groove. The form is shorter from the compact the helical structure, and most RNA and DNA duplex is in this form. You see, bila dia dehydrated, dia macam kecut kan?
Yeah.
Usually helicus per turn ialah berapa? Ini 10.9 per turn.
Usually 10 base pair per
10
turn.
bases.
Yes. Okay. You can see this one. They're longer kan?
Yeah.
Compared to this
Yeah.
one. Yeah. This one longer kan? And
Yes.
longer because of what? Because it's already hydrated.
Okay.
Usually this one is commonly found in cell lah. So they favor the high water concentration. So hydrated of major groove seem to favor B form. Okay, so it's of the right-handed and now on with only 10.0 bases per turn, and the bases incline only 2 degree from the helix. Compared to the A band just now, it is 13 kan?
Yeah.
Yeah.
The bigger degree. But this
Yeah.
one, the degrees is much smaller. So B DNA has the wide major groove. Betul lah. Besar kan?
Yeah.
Compared to this
Yeah.
one. Ini kecil kan?
Yes. It's shrink.
Okay. And the narrow minor groove and its major and groove are of about the same depth. Dia punya kedalaman pun lebih kurang sama. Bila dia dehydrated. Bila dia hydrated. How about the Z? The zDNA is a left-handed. Tadi A dengan B adalah right-handed kan?
Yeah.
But this one, the Z is the one that with the left-handed with the zigzag sugar phosphate backbone that give it the name. So it has 12 bases pattern with the base inclined with 8.8 degree from the helix. So dia macam in between lah. A ialah 13 degree. Yeah, and B is a 2 degree. And this one is between the 8.8. So zDNA has a deep major groove. Very deep one. Alright, sorry, the deep minor groove and the shallow. Dia punya shallow. Agak apa tu orang kata? Shallow mean cetek. Okay? Not deep lah. Okay? But the minors is deep. Okay? So this existence in living cell has not been proven. Okay? Then can be found in vivo or given proper sequence and superhelical tension, but function remain obscure. So kita belum tahu lagi kenapa dia ada fungsi macam ni. Okay? So this is the comparison of the 3 different forms of DNA. Okay, that you can see. Okay, you can you don't have to memorize this lah. This is just to show you the different features of the different forms of DNA structures. Okay. Now, done with DNA. Now we look at the RNA. So RNA molecule has produced by copying part of the nucleotide sequence of the DNA into a complementary sequence in RNA. So DNA make RNA in a process called transcription. So during transcription, the RNA polymerase will bind to DNA and separate the DNA strand. Okay, because DNA is double-stranded kan? Correct? So the RNA polymerase will bind and separate them into 2 different individual strands. So RNA polymerase and then use one strand of the DNA as a template from which the nucleotide are then assembled into the strand of the mRNA. Okay? Apa tu mRNA?
Messenger.
Okay. Messenger RNA kan?
Yes.
Correct?
Yeah.
Yes, doctor.
Where does mRNA being produced? Dekat mana?
In the nucleus.
In the nucleus kan? Okay? Betul?
Yes, doctor.
Okay, that one is transcription kan?
Yes.
Okay, transcript and menjadi mRNA kan? Tapi untuk translation, where does it take place?
In the ribosome, right, doctor? In the ribosome.
Ribosome dekat mana?
Endoplasmic reticulum.
Endoplasmic reticulum.
Rough endoplasm.
ER tu dekat mana?
Cytoplasm.
Cytoplasm. Betul? So,
Yeah.
mRNA, bila dia hasilkan mRNA is a very complicated punya strange, you know, because dia ada pre-mRNA. Maksudnya dia have to go through the RNA processing. Do you understand? Ataupun you belum faham, you belum belajar ini lagi? Yeah. You know mRNA is produced kan?
Yes.
Yeah.
Okay. apabila mRNA dihasilkan, the first one yang dihasilkan itu what we call is the pre-mRNA. Yes?
Yes.
Yeah. So you need RNA processing. Kerana mRNA yang kamu the pre-mRNA itu dia masih contain intron lagi. You do you understand intron and exon?
Yes.
Yes.
Only the exon
Yeah.
will be translated into the protein.
Yeah. Yeah. So the first mRNA that's produced what we call pre-mRNA because they the pre-mRNA contain introns. So the RNA processing will help to excise the intron. Can you imagine? Excise, and then they join all the exon together. Okay, tak apa. Nanti pada chapter yang lain kita akan belajar ini in details lah. Okay, so probably a lot of you don't know this, tapi tak apa. Okay, so mRNA is produced in our nucleus and it will migrate or diffuse out from the nucleus pore to cytoplasm, whereby translation will take place later. So translation occur in the cytoplasm. Make sense or not? Boleh faham ni kah?
Yes.
Boleh? Okay, so dalam synthesis protein, we have three main processes, which is replication, transcription, and translation. Betul tak?
Yes, doctor.
Yes.
Yes.
Okay, so
Yes, doctor.
transcription and replication take place in nucleus, whereby translation take place in
Cytoplasm.
okay, cytoplasm. This one you must remember. A lot of people, they don't remember. They don't understand this. Okay? Boleh ya?
Okay. Yeah, okay.
Right? Okay, good. So RNA structure is very similar to that of the DNA. So it is the polymer of the ribonucleotides. The sugars is ribose rather than deoxyribose. Okay? Senang saja. Okay? So you tengok dia OH group pada karbon 2, tapi kalau DNA dia H group saja, hydrogen saja kan. So three of its bases are the same, A, C, and G, but the U, it contains U rather than T. That means T replaced by uracil. So DNA has 4 bases, which is ATCG, but RNA that has 4 bases, which is AUCG. Okay?
Okay, doctor.
Okay, and RNA is single-stranded, but internal base pairing can produce secondary structure in the molecule. So some viruses use RNA for the genome, and in some of it, it is the double-stranded RNA, while in others, it is the single-stranded RNA, and double-stranded RNA is structurally very similar to double-stranded DNA. So in viruses, some of the viruses, they only have single-stranded. Okay, but some they have double-stranded, but you don't need to be K. You don't have to K which virus they have this. Okay, but just let you know that we have RNA can appear in single-stranded or double-stranded. Okay, so 3 main types, which is the messenger RNA, the mRNA, which transfer the DNA code to the ribose for translation. So this is the one, what we call transcript. mRNA is transcript. Okay, it's a sense strand. Sense strand that is the one that making the protein, or the one that will be translated into protein. Yeah, and we have transfer RNA, which is the tRNA, that's bring the amino acid to the ribose for the protein synthesis. Tahu tak structure tRNA macam mana? Masih ingat tak?
Tak tahu.
Ha?
Only contain exon.
tRNA. Dia bentuk dia macam clove leaf kan? Di mana dia ada anticodon kan?
Oh, yes.
Ya.
Yes.
Yang codon itu biasanya terdiri daripada 3 amino acid, betul tak?
Yes.
Ya.
Ah, masih ingat tak bagaimana transcription berlaku? Dan translation berlaku? Ya, dah lupa? Tidak apa, tak apa. Nanti kita ada spesifik tu lah. Nanti I don't want to confuse you nanti. Okay? When come to translation, transcription and translation nanti, dan kita akan bincang dengan lebih mendalam lah. So this one saya tak nak confuse you lagi. So satu lagi ialah ribosomal RNA, which is RNA that's made of the RNA and protein. Okay? Yang RNA ni yang penting untuk translation. Okay? So other RNA structure is the single nucleotide RNA or small nuclear RNA, form the complex with the protein used in the eukaryote RNA processing, like exon splicing and intro removal. This is what it calls small nuclear RNA. And we also have microRNA, a small interfering RNA, which is very short, approximately about 20 nucleotides, RNA sequences that bind to the UTR. UTR means untranslated regions, target of the mRNA, and result in the gene silencing. So a lot of methods, okay, like RNA or siRNA is being used to do gene silencing. We want to silence the gene so that we can understand the function of the gene. And we silence the gene, that means the protein will not be produced. So we want to see what happened to the organism, what phenotype that we can observe. Or this also what we call gene silencing technology. Yeah, because each gene they have their own function, kan?
Yes.
So if you want to understand the function of the gene, you need to inactivate the function of this gene, kan? How do we inactivate it? Is by silence it or knock the gene out. Pernah dengar belajar gene knockout? Okay, gene knockout, gene silencing, semuanya sama lah dia punya fungsi di mana dia nak silent the gene. Silent the gene maksudnya gene tu tidak lagi berfungsi dan protein tidak lagi dihasilkan. Boleh?
Boleh, doktor.
Boleh kan?
Boleh.
So this is the differences between DNA and RNA. So you can see the differences, kan? Ini saya tambah lah, dulu tak ada. So you can see that the full name, fungsi dia macam mana, structure dia, what's the length, sugar on it, okay? And also the bases, okay? The base pair, with the location, the reactivity, and also ultraviolet sensitivity. Okay, so these are the differences between RNA and DNA. I believe some of these features you have learned before. Okay? Boleh? Kan? Dulu
Ya.
sebahagian daripada dia punya ciri ni kamu sudah belajar kan?
Yes.
Yes,
Tapi ya, ada juga yang you belum belajar daripada ini kan?
Yes.
Okay? So dia mendalam sikit lah. Saya bagi you all. Okay? So boleh faham lah?
Yes,
Yeah.
Doctor.
Yes.
So far clear.
Clear, ha? So, ini LU3 kita lah. So, any question?
Doctor.
Yeah.
I was wondering why the DNA is spiral, not straight.
Because of the bonding.
Bonding between what?
There are so many bonds, you know. Kita ada covalence punya bond kan?
Yeah.
Kita ada hydrogen bonds kan?
Yeah.
Kita ada van der Waals bond lagi tau dalam sana. Ya? Sebenarnya ada 3 tau. Cuma kita tak bincang dengan mendalam. So ketiga-tiga bond ini akan menghasilkan satu daya, we call it kan? Force.
Okay.
Okay, forces kan? So forces yang mempunyai daya yang berbeza, dia tidak mungkin menjadi satu permukaan yang licin. Betul tak?
Yeah.
Ya, because ada satu yang kuat, ada satu yang lemah. So dia tidak mungkin hasilkan yang, unless kalau you nak licin, semua dia punya forces adalah sama.
Faham.
Make sense or not?
Ya.
Betul tak?
Betul.
Macam kita buat satu kumpulan lah. Okay, kita buat satu kumpulan di mana kita macam berpegang tangan. Ya, berpegang tangan. Dan semua orang gunakan daya dia yang paling kuat. Tapi kita kena tahu individu punya daya tu mungkin saya kuat. Kekuatan saya berlainan dengan orang kan?
Ya.
Ya, adakah kita akan menghasilkan satu barisan yang lurus?
No.
Nah, tidak mungkin. Kerana setiap orang mempunyai daya yang berbeza. Jadi dia akan bengkak-bengkok.
Okay. Faham.
Sama lah. That's why yang sama dengan DNA lah. Dia dapat helix.
Okay. Thank you, Doctor.
Okay. So, any more question?
Doctor, I have a question.
Sorry. Okay.
I asked first, I'm sorry.
It's okay.
Just now, the slides before the last second slide, the microRNA, there is a UTR target there. Just now, Doctor mentioned is target region.
Untranslated.
Oh, thank you, Doctor. Sorry,
Untranslated region. Because not the region will be translated. Okay? That is why you call 3' UTR. We have 5' UTR. Where these regions are not, will not be translated into protein.
Oh.
But this region, they may contain a very important information, which may contain enhancer.
Oh.
DNA enhancer or RNA enhancer, or they may contain the what we call translation start site in this region. So when we silence this region, that means nothing to initiate it. Nothing to initiate the translation. So translation will not be occur.
Oh, okay. Thank you, Doctor.
Yeah, make sense or not?
Yes, makes sense.
Macam satu gerabak kereta api lah. Ya. Macam steam punya kereta api, you tahu tak? Okay. Jadi dia punya engine di depan kan?
Yes.
Kalau kita putuskan gerabak depan tu, tidak ada steam, tidak ada daya, maka gerabak-gerabak di belakang tidak mungkin akan berjalan, betul tak?
Yes.
Nah, macam ni lah dia punya konsep.
Oh.
So kita silent yang di depan tu, maka yang belakang tu, the translation will not occur and also protein will not be produced. When no protein produced, that means the gene has been silenced.
Oh, okay.
Yeah, and this is how we study the function of the gene by looking at the phenotypes later, where we call gene knockout. In second year later, when you come to my developmental biology, I'll show you this in detail.
Okay.
Okay, because I've done a lot of this in my PhD study.
Okay, thank you, doctor.
All right, any more?
Yes, doctor, I have a question.
Mhm.
The snRNA and the
Hmm.
miRNA, those are new, like I've never heard that before. Can you explain a bit more about it?
Okay, this one, this SN is a small nuclei. This is a very unique one. Used in eukaryote. You only found in eukaryote. They use in exon splicing and intron removal. Because you should know that, yeah. When you remember the pre-mRNA,
Yeah, the pre-mRNA you mentioned.
yeah. Because when you do replication, right, you will replicate or you transcribe when you do during the transcription. Okay, transcription, they will transcribe intron and exon together, right?
Yes.
Okay, but intron does not contain any information. They need to be removed, right?
Mm-hmm.
Right?
Yes,
So
doctor.
the small nuclear RNA will come, help you
Oh,
to splice it out. You kena potong
Oh,
intron
okay.
itu, and then you remove the intron. So what you left with is the exon kan?
yeah.
So exon need to be annealed. Dia kena cantum sekali, ya, untuk menghasilkan mature mRNA.
Okay, now I get it.
Okay, so this mature mRNA will diffuse out from the nucleus pore to the cytoplasm.
Yeah.
And ready for translation.
And the
Okay.
miRNA is the one just now you explained to Fang Qing, I'm not sure.
Yeah, it's Fangqing.
Yeah.
What was that?
No, just now you already explained about the miRNA just now. Yeah, I understand. Thank you,
Okay.
doctor.
All right. So if you have no any question, then can we take what time is it now?
Almost 3.
Almost 3.
1
So
minute.
we take 15 minutes rest.